Usually when we talk about botanical structures, we tend to think of them in terms of passive growth.
Right. Very slow background process.
Yeah, exactly. Like a leaf slowly unfurling over a week or a vine just creeping up a trellis inch by inch. We're sort of conditioned to think of plant biology as the silent thing.
I mean, it's entirely understandable. The timeline of botanical life just doesn't usually operate at human speeds. We don't sit and watch a tree grow.
Because it's so gradual. But then, and this is what we're getting into today, you bite into a finger lime. Citrus australasica.
Oh, the citrus caviar.
Right. You experience that signature pop. It bursts in your mouth with this instantaneous, tiny acoustic snap. And I'm making a very bold claim right out of the gate here today.
Right.
That pop isn't just some fun culinary novelty. It's actually one of the most sophisticated, high-pressure liquid delivery mechanisms in the entire plant kingdom.
It really is.
It is an absolute masterpiece of cellular physics, engineered by millions of years of evolution. And honestly, it leaves our absolute best human microencapsulation technology completely in the dust.
It genuinely does. I mean, when you strip away all the gastronomy, the high-end restaurant plating, what you are actually observing is an array of independent, highly pressurized hydraulic vessels.
Yes.
These microscopic structures are managing internal geometry and pushing the absolute material limits of organic matter, with a precision that aerospace engineers spend their entire careers trying to achieve.
Okay, let's unpack this. How does a tiny fruit build a pressure vessel that rivals modern engineering? Because that is exactly our mission today.
Pulling out the microscope.
Exactly. We are stripping away all the culinary fluff, all the recipe talk, and we are focusing entirely on the cellular physics, the extreme hydraulic forces, and the structural architecture.
The things that make that explosive botanical burst actually possible.
So to you, the learner listening right now, grab your lab coat, pull up a stool, and join us. We are going incredibly deep into the biomechanics of the finger lime today.
We really need to investigate the foundational structural differences of these cells, like the osmotic engines actively driving the pressure.
And the specific mathematical equations governing the containment walls, right?
Yeah. And finally, how nature's ancient design just ruthlessly exposes the thermodynamic flaws in our modern synthetic molecular gastronomy.
Okay, so let's start at the very beginning of the timeline. How does a tiny, thumb-sized fruit from the understory of subtropical Australian rainforests build this pressure vessel?
Well, to understand how it builds the vessel, we have to understand why it's so fundamentally different from, like, the citrus fruits we eat every day.
Like regular oranges.
Exactly. The evolutionary divergence is the core of this entire structural marvel. If we look at the taxonomy, the finger lime belongs to a specific subgenus called Microcitrus.
Okay, Microcitrus, which sits within the broader citrus family, Rutaceae.
Right. And to truly appreciate what the finger lime is achieving on a biophysical level, you have to contrast it with conventional Hesperidia.
Hesperidia being just the botanical term for a modified berry with a leathery rind.
Right. Things like your standard sweet oranges, lemons, or grapefruits.
Okay, so let's visualize that standard orange. It's basically a nested, highly segmented sphere. You have the outer colorful skin, the flavado.
And then the spongy white layer just inside that, the albedo.
Yeah. And then the fruit is divided into those wedge-shaped segments by thin membranes, the septa. And inside those wedges, you have the actual juice vesicles.
Exactly. And in a traditional orange or grapefruit, let's look at the shape of those juice vesicles. They're typically teardrop-shaped.
Or kind of club-shaped, right?
Yeah. Because they develop inside those tight, wedge-shaped segmental membranes, they're densely packed. They are physically compressed into one another, filling every available millimeter.
Just squished in there.
Right. And crucially, they are bound together by a highly adhesive pectin matrix.
Wait, so when I look at an orange segment, those individual little juice sacs aren't just sitting next to each other. They're physically glued together.
Glued is the perfect word for it. Pectin acts as a biological cementing agent.
Wow, okay.
Because they're glued together in this dense, nested geometry, they share all their mechanical loads. They basically function as a single contiguous mass of tissue.
So when you apply shear force to a segment of a normal orange-like, slicing it with a knife or biting it, the tissue responds by ripping.
Exactly. The individual vesicles literally tear open across their elongated bodies. The juice just flows out in a relatively unstructured, continuous stream.
Because they're designed to fail under shear stress.
Right. But the finger lime went down a totally different evolutionary path. It abandoned the glue.
It completely abandoned the glue.
Yes, and it altered the geometry. Citrus australasica evolved to minimize almost all intervesical adhesion.
So its juice vesicles aren't teardrop-shaped. They don't taper to a point to fit into a wedge.
No, they're highly regular, distinct spherical or ovoid structures. And because they aren't bound by that sticky pectin matrix, when you cut open a finger lime, there's practically no internal pith holding them back.
They just spill out freely.
They literally look like little translucent caviar pearls just rolling out of a dark casing.
And this unglued spherical geometry changes their mechanical behavior entirely, doesn't it?
Completely.
Yeah.
When they are exposed to sheer force, again, a knife or a tooth, they don't rip. They just roll out of the way.
They evade the sheer force by moving.
Exactly. The only way you can physically breach them is through direct, high-level, targeted compression.
You literally have to isolate them and crush them.
Right.
Which brings up a huge question for me. If they aren't clumped together and sharing resources like a normal orange segment, how are they forming these perfect, independent little spheres in the first place?
Well, the developmental timeline starts incredibly early, within 1 to 2 months post-anthesis.
Anthesis being the period right after the flower fully opens and fertilization occurs.
Right, so very early on, these tiny vesicles originate as microscopic projections from the endocarp.
The endocarp is the absolute innermost layer of the fruit's peel, facing the empty cavity inside.
Correct. The epidermal and subepidermal cells in this highly specific layer begin to undergo rapid, localized anticlinal and periclinal divisions.
Okay, let's unpack those terms for the learner. Anticlinal and periclinal divisions. That means the cells aren't just multiplying in a single flat sheet, right?
Think of it like an expanding sheet of rubber. Anticlinal division means the cells are dividing perpendicular to the surface of the tissue.
So that increases the surface area, allowing the layer to expand outward.
Exactly. And periclinal division means they are dividing parallel to the surface, which adds distinct new layers of depth.
Oh, so by combining both, the tissue doesn't just get wider or thicker, it actively bulges outward into the empty three-dimensional space of the ovary locule.
It's essentially manufacturing a 3D bubble out of a flat wall. And as they grow, they differentiate into 2 distinct structural parts.
You get the main vesicle body, the spherical pearl itself, and a stalk that anchors it back to the inner wall.
Right. But there's something about this stalk that is truly bizarre. You're probably thinking about the vascular connection.
Yes, this is mind-blowing. Because when I think of a plant growing a piece of fruit, I picture it being directly plumbed into the plant's main system.
The xylem and phloem.
Right. The xylem carries water, the phloem carries sugars. It's like a house hooked up to the municipal water main. But the finger lime vesicle completely severs its direct vascular connections very early on.
It is a profound physiological adaptation. If you trace the main vascular bundles, they run robustly through the outer peel and the central core of the fruit.
But they abruptly terminate at the albedo.
Yes. They do not cross over, they do not penetrate the juice sac stalk, and they certainly do not enter the vesicle body.
Okay, I have to challenge this because I'm comparing this non-vascular stalk to, like, an umbilical cord on a space station.
Okay, I like that analogy.
If the astronauts sever the main vascular pipeline, the high-pressure hose delivering all the oxygen and water, doesn't the station die?
You would think so.
Right. If the vesicle cuts off the phloem and xylem, how does it not just starve to death? How can it possibly accumulate enough liquid and massive amounts of sugar to create that high-pressure pop without a direct pipe attached to it?
That is the core paradox of the finger lime. And the answer lies in the massive difference between bulk flow and facilitated diffusion.
Okay.
By severing the direct, high-pressure vascular pipe, the plant forces all of the transport to occur locally, either symplastically or apoplastically.
Symplastic, meaning transport through the actual living, interconnected cytoplasm of the plant cells.
Exactly, through incredibly tiny microscopic channels called plasmodemata. They link the internal fluids of adjacent cells in the stalk.
And apoplastic, meaning transport through the porous, non-living space of the cell walls outside the actual cell membranes.
Right. So instead of a high-pressure hose blasting raw sap straight into the vesicle, you have a highly regulated, much slower diffusive feed.
Just seeping through this slender, non-vascular stalk. But why go through all that trouble? Why build a bottleneck on purpose?
It seems counterintuitive, right? It would just slow down the growth.
Yeah.
It is a bottleneck, but it's a highly strategic one. This severe vascular isolation is the exact mechanism that allows the vesicle body to become an extreme chemical sink.
Oh, interesting.
Let's imagine the alternative. If the vesicle were directly plumbed into the main vascular system, its internal chemistry would constantly try to equalize with the rest of the tree.
Ah, right. It would be incredibly difficult for the vesicle to aggressively hoard extreme concentrations of solutes like citric acid because the gradients would fight to wash back into the main plant.
Exactly. It's like building a highly pressurized, sterile containment room off a small, regulated airlock.
Rather than trying to pressurize the entire space station at once.
That is a brilliant way to conceptualize it. By isolating itself at the end of this non-vascular stalk, the vesicle builds an isolated microenvironment.
Where the normal rules of vascular homeostasis just no longer apply. It can hoard whatever it wants.
And we actually see fascinating variations in the spatial geometry resulting from this exact setup, where the vesicle develops inside the fruit dictates how long that stalk gets.
Yeah, the botanical papers refer to superior and inferior vesicles.
Yes, which relates entirely to space constraints. Superior vesicles are typically found in the equatorial region, the middle of the fruit.
Because the fruit is widest there, they have a bit more room to expand outward.
Exactly. So they tend to be slightly more elongated and feature much longer anchoring stalks, sometimes as long as the entire vesicle body itself.
And inferior vesicles.
They develop near the polar ends of the fruit, the tips, where the internal space is severely cramped. They are shorter, blunter, and have minimal, almost non-existent stalks.
But whether they are superior and long or inferior and stubby, they share this fundamental blueprint. They are all vascularly isolated chemical hoarding phonolities.
Yes. And that profound isolation sets the stage for the next phase. Because it's isolated, the cell can safely manipulate its internal chemistry to weaponize water pressure.
Weaponize water pressure. Wow.
We really need to step inside the cell and look at the mechanics of cellular hydraulics here.
Okay, let's shrink down and step inside the pearl. We've established its hoarding chemicals. What does the microscopic anatomy of this vessel actually look like?
If you zoom in on the cross-section, the vesicle body is defined by a very thin, semi-rigid epidermal boundary layer, the skin.
Okay.
Inside this boundary are the internal parenchyma cells. And the absolute defining feature of these internal parenchyma cells is their vacuoles.
Okay, I remember learning about vacuoles in high school biology. It's the central storage sac of the plant cell, right? Where they keep extra water and waste.
That's the basic textbook definition, sure. But in the finger lime vesicle, the vacuole isn't just a passive storage sac. It is a monstrous structure that completely dominates the entire cellular architecture.
How big are we talking?
During the rapid cell expansion phase of the fruit's growth, these central vacuoles swell to astronomical proportions. They eventually occupy between 90 and 95% of the total cellular volume.
95%?
Yes. The actual working cytoplasm of the cell, the nucleus, the mitochondria, all of it is pushed to the absolute extreme periphery, pressed into a micro-thin layer flat against the inside of the cell wall.
So 95% of the cell is just this giant fluid-filled balloon taking up all the space. And the chemistry inside that fluid is intense, right?
Intense is putting it mildly. This is where the sheer physics of osmotic potential takes over. The vacuole actively hoards solutes.
It's pulling in simple sugars like sucrose, glucose, and fructose.
Yes, but even more importantly, it is hoarding staggering amounts of organic acids. Overwhelmingly citric acid.
I was looking at the data in our sources, and it mentions that citrate accounts for roughly 90% of the total fruit acidity in these tissues. That is a massive single chemical payload.
And as the cell aggressively pumps all that citric acid into the central vacuole, the pH drops drastically. We're talking about internal vacuolar environments reaching as low as 2.0 to 2.5 on the pH scale.
To put that in perspective, that is roughly the acidity of pure lemon juice or human stomach acid.
Right. Now, to manage this hyperacidic toxic environment and prevent it from destroying the rest of the cell, the tonoplast, which is the specific membrane that surrounds the vacuole, has to do some heavy lifting.
It uses these highly sophisticated molecular proton pumps. The primary workhorse here is something called a V-type H plus ATPase.
Exactly.
Okay, V-type H plus ATPase. We cannot just gloss over a term like that. Can you explain to me mechanically what that pump looks like and what it is actually doing?
Let's break it down. ATPase tells us that this is an enzyme that breaks down ATP.
Which is the basic energy currency of the cell.
Right. The H plus refers to a proton, a positively charged hydrogen ion. And the V-type refers to its location, the vacuolar membrane.
Okay, so picture a microscopic rotary-style motor embedded right into the membrane wall of the vacuole.
Yes. This motor literally burns cellular energy ATP2 constantly, mechanically shove protons from the cytoplasm into the vacuole, and is doing this against a massive concentration gradient.
It's forcing acid into an already overflowing hyperacidic balloon. That seems exhausting.
It's completely fighting nature. It takes immense energy to push a proton into a space that is already packed tightly with protons, but it has to do it to maintain that extreme pH.
And the citrate actually acts as a biological buffer, right? Binding to some of those free protons as they accumulate, which provides the driving force for the pumps to keep pulling even more in.
You've got it. But the ultimate physical consequence of hoarding all this sugar and all this acid is an extreme drop in what we call osmotic potential.
Which is denoted in biomechanical models as psi sub S. Let me make sure I have this right. Osmotic potential basically measures the concentration of stuff dissolved in water. The more stuff you have, sugars, acids, solutes, the more negative the osmotic potential becomes. It creates a chemical vacuum.
That is an excellent way to describe it. It's a chemical vacuum. And one of the fundamental laws of thermodynamics is that water always wants to flow from an area of higher potential to an area lower potential.
So it wants to flow from the relatively dilute watery space outside the cell straight into the hyperconcentrated acidic soup inside the vacuole.
Exactly. Because the vacuole is hoarding all this solute payload, it acts like a giant magnet for water. It pulls water across the semi-permeable membrane just through sheer osmotic force.
And as the water inevitably rushes in to try and dilute all that acid, the physical volume of the vacuole increases. It swells.
But remember, the plant cell is encased in a semi-rigid primary cell wall. The vacuole cannot expand infinitely.
Right, there's a physical limit.
As it swells, it pushes that thin layer of cytoplasm outward, directly against the unyielding cell wall. This generates an immense outward-pushing hydrostatic force. We call this turgor pressure.
And this is denoted as psi sub P.
Yes.
This is where we hit the core equation of plant biology. Total water potential, denoted by the Greek letter psi, equals the osmotic potential psi sub S plus the turgor pressure psi sub P. So psi equals psi sub S plus psi sub P.
That equation governs everything. The cell is perfectly balancing two opposing forces.
The chemical desire of water to enter, the negative osmotic potential, is physically fighting against the mechanical resistance of the cell wall pushing back the positive turgor pressure.
Exactly. And when these two massive forces finally equalize, the flow of water stops, and the cell sits in a state of extreme, high-tension thermodynamic equilibrium.
It is fully dangerously pressurized.
And because those internal parenchyma cells are so thin-walled and highly flexible, they deform easily under that pressure.
Which means they don't hold the force themselves. They transmit all of that outward hydrostatic pressure directly to the outermost boundary, the epidermal cell layer of the entire vesicle.
This is the biomechanical crux of the finger lime's entire existence. The whole spherical vesicle acts exactly like a macroscopic, thin-walled pressure vessel.
When you say pressure vessel, I am picturing industrial structures, like a propane tank for a grill or the pressurized titanium fuselage of a deep-sea submarine.
The physics are identical, whether it's a titanium submarine holding pressure out or a finger lime holding pressure in. Because it acts as a thin-walled pressure vessel, we can apply standard human engineering equations to understand the exact forces tearing at the walls.
Okay, this is wild.
Specifically, biomechanics researchers look at the tensile stress equation for a sphere.
Okay, I am looking at the notes you brought from the research papers, and I see the equation, but I am a bit lost. It says sigma equals P times R divided by 2T.
Right.
Can we walk through these variables slowly? I want to make sure I truly understand what this means in physical space.
Let's ground it in reality. Imagine holding one of those tiny, pressurized green pearls between your fingers. The Greek letter sigma, that represents the average tensile stress.
Meaning what exactly?
This is the actual physical tearing force being experienced by the skin of the pearl. It's how hard the wall is being stretched apart.
Okay, so sigma is the stretching force. What is P?
P is the internal pressure. This is that massive outward turgor pressure we just discussed, the water desperately trying to burst out.
Got it. And R is the internal radius of the sphere, basically how big the pearl has grown.
Correct. And finally, you divide all of that by 2T, where T represents the thickness of the containment wall, the epidermal skin.
So the stretching force sigma equals the water pressure P multiplied by the size of the pearl R divided by twice the thickness of its skin, 2T.
Got it.
Okay, let me look at this algebraically. If I have a fraction where R, the radius is in the numerator on top, that means as the radius gets bigger, the resulting stress, sigma, increases. The math says that as the pearl grows larger, the stretching force on its wall increases linearly.
That is the inescapable mathematical reality.
Wait, but if a pearl is bigger, shouldn't it be stronger? This math implies that the largest finger-lime pearls, the huge ones that look the juiciest and most appealing to eat, are actually the ones experiencing the most structural stress.
They are?
They're closest to exploding simply because they are big.
What's fascinating here is you are exactly right. You've hit on a fundamental law of physics. For a given internal pressure and a given wall thickness, a larger sphere will always experience significantly higher tensile stress tearing at its walls than a smaller sphere.
Wow, so just by successfully growing, by hoarding more water and getting bigger, the finger lime is actively pushing itself closer and closer to the brink of catastrophic structural failure.
It is living on the edge of its own physics. The larger the sphere, the more profound the danger.
That's terrifying for the plant.
Right. Because of those physics, the plant has no choice but to respond structurally. If the radius is increasing and the turgor pressure remains immense because of the acidic osmotic gradients, it has to do something.
The only way the plant can prevent the tensile stress from exceeding the absolute breaking point of the cell wall is to manipulate the bottom of the equation. It must engineer the thickness.
Exactly. It has to build an incredibly precise, flawless containment wall.
Because if it doesn't, if there's even a tiny microscopic weak spot anywhere on that growing sphere, that constantly increasing stress will find it.
It will.
Oh.
It will act like a microscopic magnifying glass on that flaw, and the pearl will burst prematurely while it's still safely tucked inside the fruit on the tree.
It would weep and just destroy the fruit.
Because the physics dictate that larger pearls are under immense constant stress, the plant must manufacture a flawlessly uniform wall to prevent early leaking. Which segues us perfectly from the theoretical math of the pressure to the actual physical material science of the epidermis holding it all together.
All right, let's talk about the materials. Because if I am tasked with building a sphere that holds back intense hydraulic pressure, I'm reaching for steel or carbon fiber or Kevlar. What materials is the citrus plant using to build this containment shield?
The epidermal cell layer is a breathtakingly complex biological composite material. It isn't just one thing.
Right.
It is reinforced by a thick outer cuticle layer, a highly hydrophobic epicuticular wax coating, and an underlying structural matrix comprised of cellulose, hemicellulose, and pectin.
When I hear composite material with rigid parts and flexible parts, I immediately think of fiberglass. In fiberglass, you have these rigid glass threads that provide massive tensile strength so it doesn't pull apart, but glass is brittle.
Yeah, very brittle.
So you embed those threads in a flexible epoxy resin that allows for some bend and elastic strain without shattering. Is the plant doing the same thing?
That is an exceptionally accurate mechanical analogy. In the finger lime wall, the cellulose microfibrils act exactly like the rigid glass threads. They are incredibly strong under tension.
And the pectin acts as your flexible epoxy resin, allowing the wall to stretch slightly as the turgor pressure fluctuates.
Exactly. Finally, the epicuticular wax acts as the waterproofing sealant over the whole structure to prevent that high-pressure water from simply evaporating or weeping out through the porous cell walls.
And we don't just have to guess about how strong this biological fiberglass is. Our sources include specific benchmarking data that highlights just how sophisticated this manufacturing process is.
Yes, specifically researchers Joseph Nastasi, Eleanor Hoffman, and their colleagues. They've done incredibly rigorous biophysical evaluations of different fingerline cultivars.
Their methodology is fascinating. They didn't just look at these under a microscope. They took these individual live pressurized pearls and placed them into advanced texture analyzers to map out their precise force strain profiles.
Let's explain what a texture analyzer actually is for the learner, because it sounds like something from a high-tech materials testing lab.
It essentially is. It is exactly that. Imagine a microscopic hydraulic press. The texture analyzer takes a single vesicle, places it on a perfectly flat metal plate, and lowers a highly calibrated compression probe down onto it.
And it measures, down to fractions of a millinewton, exactly how much downward force is being applied.
Right, and it simultaneously measures exactly how much the pearl deforms or squishes the strain before it finally ruptures. They crush them with microscopic precision to see exactly when and how the biological fiberglass breaks.
And their data, which compares commercial cultivars like Emerald, Champagne Red, and Chartreuse, is wild. Let's focus on the Emerald cultivar because I think it proves your point about size and stress perfectly.
Yeah, Emerald is known for producing visually striking, very large green pearls.
So, relating back to our equation, it has a large radius,
Yeah.
a large volume.
Right. But despite its large size and high internal volume, the researchers found that its epidermal wall is incredibly, almost terrifyingly thin. We are talking about an average thickness of exactly 0.25 millimeters.
A quarter of a single millimeter holding back all that massive acidic turgor pressure.
And because the wall is so razor thin and the radius is so large, the bursting force, the actual physical threshold required to rupture the pearl, which Nastasi denotes as F2 in their textural models, is significantly lower than in other cultivars.
The emerald pearls are physically fragile. They respond to very light compression. You barely have to bite them to get the pop.
Whereas a cultivar like Champagne Red or Chartreuse has a slightly different internal geometry or a thicker wall, so they register a higher F2 value. They require a much harder, more deliberate bite to pop.
But the absolute secret to the pop itself, the reason this mechanism works as an explosive burst rather than a slow, mushy leak, lies in a specific statistical measurement hidden in that benchmarking data.
We have to look at the standard deviation of that wall thickness.
Exactly, the standard deviation. Meaning, if the average thickness is 0.25 millimeters, how much does that thickness vary as you trace the surface of the sphere? Does it get thicker on one side and thinner on the other?
Right. And for the emerald cultivar, the standard deviation of that 0.25 millimeter wall is an astonishing plus or minus a 0.005 millimeters.
Plus or minus 5 micrometers. I need to pause you there. 5 micrometers.
It's microscopic.
A human hair is like 50 to 100 micrometers thick. You were telling me the plant is maintaining a manufacturing tolerance of a fraction of a human hair across an expanding, living biological sphere.
It is near perfect structural uniformity, and from a strict biomechanical perspective, this microscopic uniformity is everything. It is the whole ballgame.
Because if an industrial pressure vessel has variable wall thickness, if it has thick, heavy spots and thin, weak spots, the internal stress automatically concentrates at the thinnest points.
Exactly. Under high pressure, those thin spots will invariably yield first. They will stretch plastically, they will warp, they will thin out further, and eventually the material will fail and leak slowly.
If a finger lime had thin spots, it would just ooze. If you bit it, the pressure would find the weak spot, open a tiny microscopic tear, and the juice would just slowly leak out into your mouth. It wouldn't pop. It would be a tiny wet sponge.
It would gradually, disappointingly deflate. But because the finger lime vesicle has a near zero variance in its wall thickness, there are zero localized weak points. The entire spherical membrane is equally strong at every single geometric coordinate.
Okay, so let's walk through the exact biophysics of the bite. What happens to the math when you actually apply a compressive force to this perfectly uniform sphere?
Well, when the sliding action of your teeth or the press of your tongue against the roof of your mouth compresses the sphere, you are actively reducing its internal volume.
And because liquids, like the water in the vacuole, are incompressible, this sudden reduction in volume causes the internal hydrostatic pressure to spike instantaneously and uniformly across the entire inner surface of the barrier.
The pressure skyrockets everywhere at the exact same moment.
And because the wall is perfectly uniform in thickness, it holds. It doesn't warp. It just holds and holds and holds, absorbing that kinetic energy from your bite and storing it as extreme elastic strain within the pectin matrix.
It holds right up until the exact microsecond the pressure exceeds the ultimate tensile strength of the cellulose.
And because the wall is equally strong everywhere, there is no single point of failure. The entire structure is forced to fail simultaneously. It results in a single, explosive, catastrophic rupture. The pop.
I am thinking of this like a master glass blower, creating a perfectly blown glass ornament. If an amateur makes a glass bauble, there might be a thin spot on one side. It's fragile.
Right.
If you press your thumb against it, it cracks quietly and cleanly right at that thin, weak point. But if a master glass blower makes a sphere that is perfectly geometrically uniform and perfectly annealed, and you squeeze it in a press, it doesn't just crack.
It can absorb a massive amount of force, and then suddenly it shatters all at once.
It literally explodes into dust. Nature is essentially blowing perfectly uniform biological glass beads, but it's using extreme water pressure instead of air and biological cellulose instead of silica.
That is a phenomenal visualization. It is this flawless natural engineering, this mastery of pressure and tolerance that inevitably begs a massive question.
Yeah.
How does our very best human technology compare when we try to reverse engineer and recreate this exact sensory effect in a kitchen or a lab.
Right, because chefs have been intensely trying to copy this for a couple decades now. If you've been to a high-end restaurant, you've probably seen fake caviar.
Oh, absolutely.
We have to pivot our deep dive to molecular gastronomy, the human attempt to synthesize the finger-line synthetic spherification.
Spherification was popularized heavily in the early 2000s, utilizing various hydrocolloids to encapsulate liquid flavors, hoping to recreate that exact pop. The most common and famous method is basic spherification.
This is where a flavored liquid, say apple juice, is mixed with sodium alginate, which is a complex polysaccharide extracted from brown algae.
Right. You take this alginate mixture, pull it into a syringe, and carefully drop it bead by bead into a chemical bath of calcium lactate or calcium chloride.
I've actually seen videos of this. The instant the droplet hits the bath, the calcium ions react with the alginate. They act like little chemical bridges, cross-linking the long polymer chains of the alginate and instantly forming a gel membrane around the droplet.
You pull it out, and you have a perfect little sphere with a liquid center. Visually, they look absolutely identical to real citrus caviar.
Visually, yes, they are a stunning replica, but biophysically, when we look at the thermodynamics, they are radically, fundamentally inferior. The synthetic spheres are, at their very core, thermodynamically unstable.
They are essentially doomed from the second they are created.
Let's break down why. Let's look at the basic spherification we just described. The fundamental engineering flaw is that the chemical cross-linking reaction between the calcium and the alginate is not self-limiting.
Right. When a chef pulls that artificial sphere out of the calcium bath and rinses it in pure water, they assume they've stopped the reaction. The outside is solid, the inside is liquid. Done.
But they haven't stopped it at all.
Not even a little bit, because the ingredients to continue the reaction are still trapped inside the sphere.
Exactly. The unreacted sodium alginate molecules trapped inside the liquid core continue to migrate outward due to concentration gradients.
Simultaneously, the trapped calcium ions that form the initial gel membrane continue to diffuse inward. This is governed by fixed laws of diffusion,
which state that particles will always move from areas of high concentration to areas of low concentration until complete equilibrium is reached.
Exactly. Let me try to use an analogy here. It's like building a brick wall to enclose a room, but the mortar you use never stops drying, and it never stops expanding inward. You finish the wall, but over the next few hours, the mortar just keeps growing thicker and thicker until it completely fills the inside of the room.
That perfectly illustrates the mechanical failure. The gel membrane thickens uncontrollably inward. Within a few hours, the entire liquid core solidifies.
It converts entirely from a delicate, high-tension, liquid-filled pressure vessel into a solid, rubbery, homogeneous gel ball.
You lose the pop mechanism entirely. You just end up chewing on a piece of flavored mush.
And what about the other method? Agar-agar spheres. I know chefs use those too, where they drop a hot liquid mixed with agar into cold oil. They don't use the calcium cross-linking reaction, so they shouldn't turn solid, right?
Well, agar-agar avoids the inward gelling problem because it relies on thermal gelation, forming a physical mesh as it cools down, rather than a chemical reaction. But this thermal mesh introduces a new, equally devastating flaw. It is highly prone to syneresis.
Syneresis, what is that mechanically?
Syneresis is the extraction or expulsion of a liquid from a gel. Over a relatively short amount of time, the long polymer network of the agar gel begins to aggressively contract. It tightens up on itself.
Oh, I see.
As it tightens, it literally squeezes the water out of its own internal structure. Agar spheres will sit on a plate and rapidly whoop their liquid away into little puddles, eventually leaving behind a shriveled, dehydrated husk.
That sounds awful.
And furthermore, the agar mesh has terribly low elastic strain. It doesn't stretch to absorb force. It just tears under very low shear forces. It doesn't pop. It just breaks apart.
Here's where it gets really interesting to me. The synthetic spheres we just talked about, alginate and agar, are basically dead chemistry running down a clock.
The second they are formed, thermodynamics starts tearing them apart or turning them to rubber.
But the natural citrus caviar, the finger lime pearl we've been analyzing, it is a living system.
That is the ultimate defining difference. It is actively alive.
It has active functioning cell membranes. It has mitochondria burning sugar and providing ATP. It has those complex vacuolar transport pumps, the VATPs we talked about earlier, constantly spinning and working.
The cell is actively, relentlessly expending its own biological energy to lock the pearl in thermodynamic stasis.
It is maintaining a highly dynamic equilibrium. The living machinery within that tiny, micro-thin layer of cytoplasm constantly repairs and reinforces the cellulose matrix.
It actively manages the harsh osmotic gradients. It actively utilizes proteins and enzymes to prevent the internal vacuolar fluid from ever gelling or weeping out.
It maintains that high elastic strain and perfect fluid compartmentalization indefinitely, for as long as it remains attached to the tree and viable.
It isn't just sitting there. It is actively fighting the laws of entropy every single second of its existence.
Let's build on that and connect this to the much bigger picture. Think about what it actually takes to build a microscopic water balloon that never loses high pressure, never turns to solid rubber over time, and flawlessly maintains a geometric manufacturing tolerance of 5 micrometers across its entire surface.
It's mind-boggling.
When you look at it that way, which engineer do you ultimately trust more to build a flawless pressure vessel, a lab technician with a plastic syringe of sodium alginate, or millions of years of rigorous, unforgiving evolutionary selection?
It's honestly not even a contest. The lab technician is playing with wet sand on the beach while evolution is building skyscrapers.
Exactly.
But I think to be completely fair to the lab technicians and chefs of the world, even nature's perfect pressure vessels have their absolute breaking points. Nothing organic is truly indestructible. What happens when the external environment pushes the internal physics of the finger lime too far?
That is a crucial area of biomechanical study. Because these vesicles are engineered to operate at such extreme high-tension limits, when the biomechanics do fail, they fail catastrophically. We can see this vividly illustrated in specific physiological disorders like Stylerin breakdown or SEB.
A SEB, which is extensively documented in closely related species like key limes, citrus, or antifolia. What environmental factor triggers that kind of failure?
It is primarily triggered by severe, acute environmental stress, most notably extreme heat events. When the fruit is still on the tree and is exposed to direct, intense solar radiation during a heat wave, the internal temperature of the fruit's tissues can spike dramatically, sometimes reaching between 42 and 50 degrees Celsius.
50 degrees Celsius is 122 degrees Fahrenheit. That is an incredibly hot temperature for a living piece of fruit to endure.
And remember the foundational physics we discussed earlier regarding the internal vacuole. Liquids expand when heated. The highly pressurized hypercitic vacuolar fluid trapped inside the vesicles undergoes rapid thermal expansion.
As the liquid expands, it demands more physical volume.
Right. And because the cell wall is rigid, this causes the internal outward pushing force, P, to surge exponentially.
And we go right back to our mathematical equation, sigma equals P times R divided by 2T. If P, the pressure, surges due to heat expansion, and T, the thickness, is completely static, the tensile stress sigma spikes violently.
The wall's being stretched far beyond its normal high-tension state.
Exactly. And compounding the problem, that extreme heat is also potentially denaturing some of the structural proteins within the membrane and softening the pectin matrix that holds the cellulose together.
So the ultimate tensile strength, key, of the cell wall may actually be simultaneously weakening while the pressure, P, is skyrocketing. Eventually, the pressure simply overwhelms the containment shield.
So the pearls pop, but they were popping while they are still deeply buried inside the fruit on the branch.
Yes, the vesicles suffer catastrophic internal ruptures. The highly acidic, concentrated juice blasts outward into the surrounding delicate albueo and flavado tissues of the rind.
This pH 2.0 acid immediately destroys the cellular integrity of the rind, causing deep, dark, necrotic lesions on the skin, essentially destroying the fruit from the inside out.
It is a cascading, unrecoverable mechanical failure.
And it's not just heat that can destroy the physics, right? Cold can break the pressure vessels too.
Indeed. Finger limes are what we call non-climacteric fruits, meaning they do not continue to ripen after they are harvested from the tree. But they are incredibly, notoriously sensitive to post-harvest storage conditions.
If you harvest them and store them at 45 degrees Celsius, which are standard commercial refrigeration temperatures, they suffer rapid chilling injury.
How does the cold physically break the vessel? Does the liquid freeze and expand?
It doesn't even need to freeze. The sustained cold physically damages the lipid bilayers of the living cell membranes. The lipids stiffen and lose their selective permeability.
Remember those VAT paste pumps? Constantly maintaining the osmotic gradients? The cold shuts them down and breaches the membrane seal.
The extreme osmotic gradients simply collapse. The turg or pressure is instantly lost, and the vesicles suffer widespread pitting and severe fluid loss.
The perfect high-tension pressure vessel just miserably deflates into a mushy husk.
That shows just how fragile the living system is to temperature. But the plant isn't completely defenseless against all stress, especially when we talk about biological threats. The sources dedicate a significant portion to discussing the finger lime's tolerance to HLB.
Huanglongbing, also globally known as citrus greening disease. It is caused by the phloem-restricted bacterium candidatus Labrobacter asiaticus.
HLB is arguably the most destructive citrus disease in recorded history. It has absolutely devastated conventional multi-billion-dollar citrus industries globally.
The bacteria multiply voraciously within the vascular tissue of the tree, literally choking off the flow of nutrients. This leads to bitter, severely misshapen fruit, massive crop drop, and eventual inevitable tree death.
Our standard sweet oranges and grapefruits are highly tragically susceptible.
But the finger lime, Citrus australasica, manages to fight back against this global killer.
It does. Extensive field experiments have shown that finger limes exhibit remarkable, robust field tolerance to HLB. When infected, they show a much lower overall bacterial titer, meaning fewer bacteria circulating in their systems, and they exhibit far fewer physical symptoms of the disease compared to oranges.
And the molecular mechanisms behind this defense are a profound testament to the active, highly dynamic nature of their cell walls.
This goes back to your point about the synthetic sphere as being dead chemistry. Unlike a synthetic alginate sphere, which just sits there passively while bacteria or mold eat it, the finger lime pearl can actively remodel its own architecture.
How does a tiny, vascularly isolated sac even know a deadly bacteria is in the neighborhood? What trips the alarm system?
It relies on sophisticated biological sensors. The plant cells contain surface receptors that can detect what we call PMPs, pathogen-associated molecular patterns.
These are essentially chemical signatures, fragments of the bacteria's own cell walls or flagella floating in the extracellular space.
When the receptors on the finger lime cells detect these bacterial signatures, it triggers a massive, instantaneous intracellular alarm cascade. And the plant responds by rapidly altering its own structural materials.
What does it do? Does it build a thicker wall?
It focuses entirely on the vulnerabilities. It begins violently depositing highly defensive, impenetrable biopolymers, specifically callus, lignin, and suberin, directly at the plasmodesmata.
Remember, those are the tiny microscopic channels in the non-vascular stalk we discussed earlier, the only symplastic pathways into the chemical sink.
So it's not just a passive water balloon holding acid, it's an intelligent reactive material. It detects an invading bacterial force, and it instantly chemically concretes over its own microscopic access doors to physically barricade the bacterial spread.
That is precisely the mechanism. Let's look at callus, for example. Callus is a highly durable polysaccharide that acts almost exactly like a fast-curing biological sealant.
By rapidly synthesizing callus and plugging the plasmodesmata in sieve pores in the stalk, the plant willingly sacrifices the metabolic transport efficiency of that stalk.
It cuts off its own slow drip supply line to prevent the systemic spread of the bacteria into the extreme isolated chemical sink of the vesicle.
It isolates the infection structurally, starving the bacteria out. It is a highly active, real-time emergency remodeling of the pressure vessel's foundation.
That is absolutely incredible. A smart, self-sealing, high-pressure hydraulic vessel that can detect pathogens and concrete its own doors shut.
So bringing this entire investigation together, let's summarize the core thesis of what we've discovered by putting this fruit under the microscope today.
If we synthesize the biomechanics, the hydraulics, the material science, and the defensive remodeling,
the finger lime conclusively proves that true material sophistication doesn't come from synthesizing new static polymers in a lab. It doesn't come from a chef playing with a syringe of algae extract.
true engineering brilliance comes from the hierarchical, highly intelligent organization of natural, actively living materials.
Human microencapsulation relies on thermodynamically doomed chemical tricks that begin to degrade the very second they are formed. Nature, however, balances extreme osmotic potentials, perfect geometric shapes, manufacturing standard deviations of 5 microscopic micrometers, and dynamic living cell walls to create a self-maintaining pressure vessel that can actively defend itself against global bacterial pathogens.
It is the stark difference between a static, decaying snapshot of human engineering and an actively maintaining, dynamically repairing living system. But before we close lab doors and wrap up today, there's one final, incredibly striking concept hidden in the physics of these vesicles that we haven't even touched on yet.
Hit me. We've covered pressure, math, and bacterial defense. What is the final piece of the puzzle?
It involves the optical physics of the finger lime. When biophysicists analyzed the bulk optical properties of these tissues, they found something deeply astonishing. Let's look at normal citrus tissues first. like the spongy white albedo or the outer colorful flavado of an orange peel. They have a very high bulk scattering coefficient.
Let me pause you there to make sure I understand. A scattering coefficient. When light hits an object, it either passes straight through, it gets absorbed, or it scatters. It bounces off the internal structures in a million different directions.
Precisely. In normal plant tissue, light enters and scatters wildly, bouncing randomly off the complex, disorganized cellular structures, the air pockets, the thick pectin matrix. Their scattering coefficient ranges from 25 to 35 inverse millimeters.
Inverse millimeters. What does that unit actually measure in physical space?
Think of it as a measurement of frequency per distance. An inverse millimeter essentially tells you how intensely or how frequently light is scattered for every single millimeter it travels through the material. A value of 35 inverse millimeters means the light is bouncing around violently and constantly as it tries to penetrate.
This immense scattering is what makes the orange peel completely opaque. The light just can't get through in a straight line.
Right. You can't hold a flashlight to an orange peel and see through it like glass.
But the pressurized, isolated juice vesicles of the finger lime possess a shockingly low bulk scattering coefficient. Across the entire visible and near-infrared light spectrum, their coefficient is measured at less than 3 inverse millimeters.
Less than 3 compared to a normal tissue's 35. That's not a minor variance. That's a massive foundational difference in how the material interacts with light.
And the reason goes right back to the extreme cellular architecture we discussed at the beginning. Because the massive, liquid-filled central vacuole pushes all of the dense organelles, the nucleus, the mitochondria, to the absolute extreme microperiphery of the cell. And because the cell walls themselves are so incredibly uniform, devoid of thick spots or heavy pectin glue, there is practically nothing inside the sphere for the light to bounce off of.
The internal liquid is pure. The light passes right through the entire structure almost entirely unscattered. This exact lack of internal optical interference is what gives the finger lime pearls their stunning glass-like translucency. They are optically remarkably pure.
Okay, my mind is racing with the applications of this. So we have a biological structure that perfectly dynamically contains extreme hyperacidic hydrostatic pressure. It utilizes an ultra-thin quarter-millimeter membrane with microscopic manufacturing tolerances. It can actively detect and seal out bacterial invaders. And simultaneously, as a byproduct of all this engineering, it allows light to pass through it almost completely uninterrupted.
I suggest pondering this exact convergence of traits. If modern material scientists can fully reverse engineer this highly specific biological architecture, a structure that flawlessly masters both extreme internal hydraulic pressure containment and profound optical clarity, could the finger lime be the ultimate organic blueprint for the next generation of human technology?
Could we be looking at the biological template for entirely biodegradable, liquid-filled optical lenses?
Or perhaps ultra-sensitive deep-sea pressure sensors that require perfect internal optical transparency to transmit data, built on the structural mathematics of a piece of fruit.
A deep-sea pressure sensor built on the cellular architecture of a tiny citrus fruit from the Australian rainforest. That idea, that brilliant intersection of nature and engineering, is exactly why we do these deep dives. To you, the learner, thank you for joining us in the virtual lab today. We've gone from the evolutionary severing of microscopic vascular umbilical cords through the high-tension mathematics of tensile stress, the thermodynamic flaws of fake caviar, and finally into the crystal-clear optical purity of high-pressure fluid dynamics.
It is an immense amount of cellular physics to process.
But the very next time you hear that signature tiny pop, whether it's sitting on a plate in front of you or you see it in a video, just remember, the most advanced, mathematically perfect fluid engineering on the entire planet isn't happening in a sterile, high-tech manufacturing plant. It might just be growing, quietly and perfectly, on a thorny bush.